Hello, everyone. The results that I will show today is actually based on our recent PCCP paper. This is a somewhat exotic topic because of two aspects, photo switching and metal-free activation of parahydrogen. So I think I should give a short introduction to both of these. And in general, involvement of photoactivated reactions in FIP is interesting as it adds an orthogonal way to control the hyperpolarization process. Here are some selected examples from the literature. For instance, it's possible to photoactivate ejection of hydride ligands from metal complexes using a light, which leads to highly active 16-electron intermediates. Typically, it's for ruthenium complexes. And then that active species can react with parahydrogen really rapidly. This is a laser pump technique developed by Megan Hals, Simon Duckett, and co-workers. Among other things, it allows the observation of interesting effects such as oscillations of zero quantum coherences, basically sort of quantum bits in the formed hyperpolarized complexes. Visible light activation of hydrogenation reactions can also be used to enable FIP using photosensitizers. That's his work from Dice Lab. Also, Alexei Kirutin and co-workers showed that photo switching of azobenzene stereoisomers can be used to lock nitrogen 15 hyperpolarization in the trans isomer in SABRE. Also, using SABRE, the Dice Lab demonstrated that photoactivated ejection of pyridazine and pyrazine ligands facilitates the formation of free hyperpolarized forms of these ligands. Actually, in all these examples, metal complexes served as the centers that chemically activate parahydrogen. This activation is actually very crucial since, as we all know, parahydrogen itself is an MR island. We need chemistry for FIP. In these cases, this is metal-based chemistry. At the same time, metal-free catalysts or activators for parahydrogen are also known, and my group has been studying them for quite a while. Compared to metals, metal-free catalytic systems for small molecule activations are generally considered as more sustainable and in some respects less toxic since they are based on abundant light main group elements. They include two main classes, frustrated Lewis pairs represented by various ansa-aminoboranes and ansa-phosphinoboranes. And also there are bi and tetra radicaloids, kind of another important class of such compounds that can activate parahydrogen, and we can observe hyperpolarization effects. Generally, pnictogen radicaloids, and in particular phosphorus biradicaloids, can activate parahydrogen, resulting in the formation of hyperpolarized adducts. And there are several types of such molecules, including 4- and 5-membered heterocyclic species, as well as tetraradicals. All of them share a common feature. They are open shell species in which the electron spins are in singlet states. Therefore, the radical centers do not cause any deleterious effects such as line broadening in NMR. More importantly, among these species, 5-membered biradicaloids are photoactive, and that is used in the work that I will present. The activation of parahydrogen by such 5-membered biradicaloids is an irreversible process. Here is an example of biradicaloid 1. For this compound, the resulting hyperpolarized hydrogen adduct 3 forms an kind of spin system, AMXY spin system, where all spins, I'm talking about hydrogen, phosphorus, phosphorus, hydrogen. In this case, all of them are weakly coupled. its Pasadena spectrum shows two sets of antiphase signals from two parahydrogen-derived hydrogen due to coupling with the phosphorus 31 nuclei. More importantly, this biradicaloid can be switched to inactive Hussein form by visible red light. As we showed in our communication, illumination using red light interrupts the reaction with parahydrogen through that photo switching from 1 to 2. So basically, we can stop the reaction of interaction between biradicaloid and parahydrogen by converting the first one to the Hussein form. When the light is off, in contrast, thermal back conversion regenerates the active biradicaloid, and its reaction with parahydrogen leads to hyperpolarization effects. That is basic idea. A relatively simple experimental setup was used for these experiments. An optical fiber with a Rathan tip was inserted into solution to control the photo switching, and parahydrogen was supplied through a thin capillary to reach 6 bar pressure. So we worked under elevated pressures. The sample preparation in this experiment began with initial conversion of biradicaloid 1 into inactive Hussein form. We need to switch on the light. And quite quickly, dark blue solution is converted into something more transparent, and basically, finally, it will be pale yellow color of the inactive Hussein form. Then after that, what we need to do, we need to bubble parahydrogen into that solution under illumination conditions so that we can saturate solution with parahydrogen for further experiments with actually photo switching. The next step is basically NMR experiments, and here in this video you can see that how we can switch off the light, for instance, and the same way it's possible to switch on and switch off the light when the sample tube is inside an NMR magnet. After the sample preparation, we observed a clear response to light in the proton NMR spectra. Light on periods, as it is shown here for one of the protons in the adduct of the pyradical with parahydrogen. So during light on periods, we do not observe any signal enhancement, and during light off events, we observe strong NMR signal enhancement. By the way, the absolute mode spectra are plotted here as a function of time, but normally the signals are antiphase, as I showed kind of a couple of slides before. The response to switching the light was quite clear and fast, and we did not observe any modulations of the signal amplitudes, as this is the so-called slow photo FIP. Yeah, it's nice, but of course, such photo switching events cannot continue indefinitely. During each light off period, there is a drop in the amplitude of the enhanced signals, as it's shown in this figure. Basically, that is a Pasadena signal as a function of time and during light on and light off periods. This behavior, kind of dropping the signal, is mainly determined by the irreversibility of parahydrogen addition and the gradual consumption of the active biradicaloid form in solution. This becomes even clearer if the light on periods from this graph would be removed, and we can plot some sort of decay of the enhanced signals as a function of time during light off periods, basically. The rate constant of this decay correlates well with that of the thermal back conversion from the inactive Hussein form. Basically, when the light is off, the thermally activated back conversion from 2 to 1 is the rate determining step on the way towards the hyperpolarized product, because the irreversible reaction of reaction with parahydrogen is fast. And that was shown by independent kinetic measurements of this process, and those are supporting the ideas because this idea, because the time scales obtained for the backend version without parahydrogen correlate well with the signal decay observed in the parahydrogen experiments. This slide brings me to my conclusions already. In our communication, we show that metal-free activation can be coupled with visible light photo switching. Through this coupling, hyperpolarization can be controlled by light. In contrast to metal-based parahydrogen activation, when we speak about photo activations, illumination generates inactive species in the metal-free case, effectively switching off the hyperpolarization. When we use metal-based systems, it's typically by light we generate active species that can react with parahydrogen. There is room for development. For instance, it's possible to use phosphorus 31 NMR to detect photoresponse by polarization transfer to the phosphorus 31 nuclei. Also, one intriguing direction is to use some structural modifications of that 5-membered biradicaloids to achieve reversibility of H2 addition. That is not easy to do, but that may allow to have much longer kind of duration of cycles of switch on and switch off events. And these are acknowledgements that I would like to say specifically to my colleagues from the University of Rostock, lab of Professor Axel Schulz, who are really great specialists in biradicaloid chemistry. Thank you very much for your attention.